بسم الله الرحمن الرحيم والحمد لله رب العالمين والصلاة والسلام على أشرف المرسلين سيدنا محمد عليه أفضل الصلاة وأزكى التسليم. Today our lecture is the physiology of basal ganglia. Physiology of basal ganglia. During this lecture we have to understand the functional organization of the nuclei that form the basal ganglia, their input and output and their connection to the cerebral cortex through the thalamus, the two different pathways, the direct and indirect pathways of the basal ganglia and their role in the coordination of the voluntary movement, the role of the basal ganglia in higher order motor function, the role of basal ganglia in behavioral and cognitive function, the neurotransmitter and the different neurotransmitters present in the basal ganglia, and the disturbances that associated with the neurotransmitter metabolism and the diseases affecting or the disorders affecting the basal ganglia like Parkinson's and Huntington disease. So these are the objectives that we are going to deal today. Structure of the basal ganglia. The basal ganglia are formed of group of nuclei, and these nuclei play an important role in the initiation and regulation of motor activity. The nuclei of the basal ganglia are classified or distributed as the follow: the caudate nucleus, putamen, and the globus pallidus. And the caudate and the putamen are known as the striatum, and these three nuclei are located in the telencephalon, The globus pallidus is formed of two segments, internal and external segments. The fourth nucleus is the subthalamic nucleus, and this is present in the diencephalon below the thalamus. The last nucleus is the substantia nigra, which is present in the mesencephalon, and is formed of the substantia nigra pars compacta and substantia nigra pars reticulata. So we have five nuclei. These five nuclei forming the basal ganglia: caudate, putamen, globus pallidus. The globus pallidus is formed of external and internal segments. These nuclei are present in the telencephalon. In the diencephalon we have the subthalamic nucleus, and in the mesencephalon we have the substantia nigra. These are the five nuclei of the basal ganglia which play an important role in the initiation and regulation of motor activity. Now we are going to look for the neural connection, the inputs and the outputs, or the afferent and efferent pathways for the basal ganglia. If we look here to the cerebral cortex, these areas represent the inputs to the basal ganglia. This means that the basal ganglia receives input from all the cerebral cortex except the primary visual and the primary auditory cortex. This means that the basal ganglia receives input from all areas of the cerebral cortex except the primary visual and auditory cortex. Usually the input going to the basal ganglia is entering the basal ganglia through the striatum or the caudate and putamen. So the caudate and putamen is the principal entry gate for the afferent pathway to the basal ganglia. And as we mentioned, the major input or the major component of the afferent input comes from the whole cerebral cortex except the primary visual and the primary auditory cortices. But the heaviest contribution is coming from the association areas located in the frontal and parietal cortices. The major contribution or the heaviest contribution is coming from the association areas in the frontal lobe and in the parietal lobe. And if we look to the cortico-striatal projection, this means that the fibers coming from the cerebral cortex going to the caudate and putamen or the striatum, they are named as the cortico-striatal projection. Usually they synapse with a certain type of neurons called the medium spiny neurons, and these neurons are GABAergic neurons. So the corticostriatal fibers coming from the cerebral cortex synapsing with the caudate and putamen are synapsing with the medium spiny neurons of the striatum, which is GABAergic neurons. And if we look to the caudate receives input from multimodal association areas and from the eye movement area in the frontal lobe, while the putamen receives the input from the primary and secondary somatic sensory area in the parietal lobe, secondary visual area in the occipital lobe, premotor and motor areas of the frontal lobe, as well as the auditory association area in the temporal lobe. So most of the cerebral cortex is going to synapse or give connections with the caudate and putamen. And if we look to something called the putamen circuit or the caudate circuit, we have something called the putamen circuit and the caudate circuit. The putamen circuit. The putamen circuit. Here we have this is the medium spiny neurons, the medium spiny neurons located in the caudate and putamen, and that is the site of synapsing of the corticostriatal fibers. The fibers coming from the cerebral cortex is going to synapse with the medium spiny neurons. If we look to the putamen circuits, usually the fibers will start from the premotor area and the supplemental motor area, and then go to the putamen, and from the putamen to the globus pallidus, especially to the globus pallidus internal segment, and from the globus pallidus internal segment the fibers will go to the ventrolateral and the ventroanterior nuclei of the thalamus, and the fibers will go back to the cerebral cortex to the primary motor area. So we have something which is a putamen circuit, which is a closed circuit starting from the premotor area and supplemental motor area, going to the putamen nucleus of the basal ganglia, to the globus pallidus internal segment, to the ventrolateral and ventroanterior nuclei of the thalamus, and then back to the cerebral cortex, and then back to the cerebral cortex to the primary motor area. And if we look to the caudate circuit, that will start in the association areas, that will start with the association areas, especially the somatic and visual and auditory association areas, going to the caudate, and from the caudate to the globus pallidus internal segment to the thalamus, mainly to the ventrolateral nucleus of the thalamus, and then to the premotor area of the cerebral cortex, and then to the premotor area of the cerebral cortex. So it will end in the cortical or in the cortical motor association area. And if we look to the function of these two areas or these two circuits, the putamen circuit is concerned with the executing pattern of learned movement, while the caudate circuit plays an important role in production of the cognitive function of the basal ganglia, As we mentioned, the caudate and the putamen are known as the striatum, and the major input to the basal ganglia will enter through the caudate and putamen, and the fibers is mainly called corticostriatal fibers, arising from different regions of the cerebral cortex except the primary visual and the primary auditory cortices, while we have specific circuits. One is known as the putamen circuit and the other one is known as the caudate circuit. Efferent pathways. The fibers coming out from the basal ganglia. The fibers coming from the basal ganglia is usually coming from the globus pallidus internal segment, and usually these fibers go to the ventroanterior and the ventrolateral nuclei of the thalamus before reaching to the frontal cortex or to the motor cortex. So the globus pallidus internal segment represents the major efferent pathway or the major output pathway from the basal ganglia. Before reaching to the cerebral cortex, they have to make a synapse in the ventrolateral and the ventroanterior nuclei of the thalamus. Also to the superior colliculus from the substantia nigra pars reticulata to the superior colliculus, that is usually produced or coming from the substantia nigra pars reticulata. The superior colliculus usually produce control of the hand and eye movement. And if we look to the efferent output of the basal ganglia, it is usually inhibitory. These neurons are GABAergic neurons. They are mainly inhibitory, and they are producing the inhibitory action of the basal ganglia on the motor function. These neurons are mainly GABAergic neurons. These neurons inhibit the unwanted movement because they have high level of intrinsic activity. They usually inhibit the unwanted movement produced by the motor areas because these areas usually have high level of intrinsic activity. In addition, efferent pathways from the globus pallidus internal segment and substantia nigra pars reticulata project also to the brainstem and reticular formation, So some of the fibers will project to the reticular formation from the globus pallidus and from the substantia nigra. Again, the efferent pathway from the globus pallidus internal segment to the thalamus before reaching to the motor cortex, to the superior colliculus from the substantia nigra pars reticulata. These neurons are GABAergic neurons. They produce inhibition of the unwanted movement, and that is the main function of the basal ganglia. Some of the efferent pathway will project from the globus pallidus and from the substantia nigra to the reticular formation in the brainstem. Then we have the internal connection or the connection that is present between the nuclei of the basal ganglia. We have a pathway from the striatum to the globus pallidus, striatopallidal fibers. We have a pathway from the caudate to the substantia nigra pars reticulata, from the caudate to the substantia nigra pars reticulata, and we have a reciprocal pathway between the globus pallidus and the subthalamic nucleus. We have a pathway from the subthalamic nucleus to the substantia nigra pars reticulata, and we have the nigrostriatal fibers. The fibers that is secrete or release dopamine, and these cell bodies are present in the substantia nigra pars compacta, and the fibers end in the caudate and putamen, or end in the striatum. And the most important related to these fibers, that is the damage or the decrease the release of dopamine from this fiber, is associated with the Parkinsonism. So again, the internal connection from the striatum to the globus pallidus, from the caudate to substantia nigra pars reticularis or pars reticulata, reciprocal pathway from the subthalamic nucleus to the globus pallidus, from the substantia nigra pars compacta to the striatum, the nigrostriatal fibers, and from the subthalamic nucleus to substantia nigra pars reticularis. Now we are going to look for the neurotransmitters secreted in between the basal ganglia, secreted in between or inside the basal ganglia. We have different types of neurotransmitters secreted in the basal ganglia, and we will start with the first type, which is glutamate, secreted from glutaminergic neurons. We will find the corticostriatal neurons, coming from the cerebral cortex to stimulate the striatum, are mainly dependent on the releasing of glutamate from the cerebral cortex to striatum to excite it. Also, the globus pallidus is also receiving glutamate from the subthalamic nucleus. We have cell neurons or neurons secreting glutamate in between the subthalamic nucleus and the globus pallidus. So the first type of neurons is the glutaminergic neurons. The glutaminergic neurons include the corticostriatal neurons and the neurons coming from the subthalamic nucleus to the globus pallidus. And glutamate is the major excitatory neurotransmitter, in the basal ganglia. The second one is the dopaminergic neuron, or the second neurotransmitter is dopamine. Dopamine is secreted mainly from the nigrostriatal fiber. It is secreted in the striatum from the substantia nigra pars compacta. And dopamine, acting on the dopaminergic receptors type 1, producing excitatory effect, while when it produces stimulation of the dopaminergic receptors, produce stimulation of the dopaminergic 2 receptor produce inhibitory effect. So the dopamine secreted from the substantia nigra pars compacta to the striatum or from the nigrostriatal fiber when bind with dopaminergic receptor subtype 1 produce excitatory effect, and when it bind with the dopaminergic subtype 2 will produce inhibitory effect. So that is the actions of the dopamine. Dopamine on the dopaminergic subtype 1 will potentiate the effect of the cortical glutaminergic input to the striatum. It means that it's going to facilitate the action of glutamate, while on the D2 receptor it antagonizes the action of glutamate. So the second neurotransmitter is dopamine, secreted by the nigrostriatal fiber, excitatory on D1, inhibitory on D2. The third type of neurotransmitter is acetylcholine. Acetylcholine is secreted from some of the corticostriatal fibers and also secreted from small interneurons located in the striatum, and the acetylcholine is excitatory in the basal ganglia. The last neurotransmitter is the major inhibitory neurotransmitter, which is GABA. GABA or gamma aminobutyric acid is secreted by cell bodies located in the striatum and projecting to the globus pallidus and substantia nigra pars reticulata, and also secreted from the substantia nigra pars reticulata to the thalamus and from the globus pallidus to the reticular formation. So it is secreted from striatal GABAergic neurons that project to the globus pallidus and to the substantia nigra pars reticulata. Also, the input coming from the globus pallidus internal segment to the thalamus is mainly inhibitory through the secretion of GABA. Again, we have four types of neurotransmitters present in the basal ganglia, or four major types of neurotransmitter present in basal ganglia. Glutamate, and the glutamate is mainly coming from the corticostriatal fibers to the striatum, excitatory. We have acetylcholine, local interneurons and from corticostriatal fiber. Some of the glutaminergic fibers are coming from the subthalamic nucleus to the globus pallidus, excitatory. Then we have the nigrostriatal fibers secreting dopamine, and dopamine either produce excitatory effect or inhibitory effect according to the stimulated receptor. And if we look to the last one, which is GABA, GABA is coming from striatal neurons, which is the medium spine neurons coming from the striatum to the globus pallidus and to substantia nigra pars reticulata, and from the globus pallidus to the thalamus. Lastly, the thalamus is secreting glutamate on the cerebral cortex, exciting the cerebral cortex. This means that the output coming from the thalamus back to the cerebral cortex is excitatory through the release of glutamate. Now we are going to look for the major pathway share in the function of the basal ganglia, which is either the initiation of voluntary movement or the inhibition of unwanted movement. We have the first pathway is the direct pathway. The direct pathway facilitates the initiation of the voluntary movement. One of the actions of the basal ganglia in the regulation of the voluntary movement is mediated through the direct pathway. And this occurs when the cerebral cortex becomes stimulated, and when they are stimulated they release excitatory neurotransmitter on the striatum, on the caudate and putamen, stimulating the caudate and putamen. Once they stimulate the caudate and putamen, they secrete GABA, and this GABA is inhibitory, so this will inhibit the globus pallidus internal segment neurons. So we have inhibition of the globus pallidus internal segment. The globus pallidus is usually inhibiting the thalamus through the release of GABA. So if we inhibit the globus pallidus, we will decrease the release of GABA on the thalamus, and this will produce disinhibition of the thalamus or release of the thalamus from the inhibition. This means the thalamus will be stimulated, and the stimulation or disinhibition of the thalamus will be associated with stimulation of the cerebral cortex. The stimulation of the cerebral cortex will produce the initiation of the voluntary movement. And if we look to the action of dopamine, dopamine usually bind with dopaminergic subtype 1 receptor, bind with dopaminergic subtype 1 receptors. This means that it will facilitate the action of glutamate, it will activate the direct pathway. So we have the direct pathway, which is concerned with the initiation of, or the facilitation of the initiation of the voluntary movement. The dopamine will excite or stimulate the dopaminergic subtype 1, increasing the activity or facilitating the action of glutamate, activating the striatum. The striatum inhibits the globus pallidus internal segment, which leads to decrease the release of GABA from the globus pallidus to the thalamus. Thalamus become disinhibited, or in between brackets stimulated, and this will stimulate the cerebral cortex. Role of dopamine: facilitate or activate the direct pathway. That is the direct pathway that help in the initiation of voluntary movement. The second pathway is the indirect pathway. The indirect pathway is more complex, and usually it produces suppression of inappropriate movement or the termination of voluntary movement. The indirect pathway usually produces suppression of inappropriate movement, or we can make it or say as termination of the voluntary movement. And the condition also started with stimulation of the cerebral cortex. Cerebral cortex is going to produce stimulation of the striatum. The striatum is going to release GABA, and this GABA is inhibitory, but now on the globus pallidus external segment. It will produce release of GABA on the globus pallidus external segment, and this will decrease the amount of GABA secreted from the globus pallidus external segment to the subthalamic nucleus. And in this condition, the subthalamic nucleus become stimulated. Or disinhibited. The subthalamic nucleus release glutamate on the globus pallidus internal segment, will release glutamate to stimulate the globus pallidus internal segment. Once we stimulate the globus pallidus internal segment, it's going to inhibit the thalamus, and when it inhibits the thalamus, this will inhibit the cerebral cortex. So again, excitation of the cerebral cortex associated with stimulation of the striatum increase the release of GABA on the globus pallidus external segment. This will produce decrease or less GABA released from the globus pallidus external segment to the subthalamic nucleus. Subthalamic nucleus become disinhibited, or in between brackets stimulated, releasing GABA on the globus pallidus internal segment. Stimulation of the globus pallidus internal segment release GABA to inhibit the thalamus. Thalamus will inhibit the cerebral cortex. So once we inhibit the cerebral cortex, we produce suppression of the inappropriate movement, or we will produce termination of the voluntary movement. And if we look to the action of dopamine is inhibitory on the striatum through the D2 receptors. Dopamine will antagonize the effect of glutamate on the striatum, so it will decrease the activity of the indirect pathway. As it antagonizes the effect of glutamate here, it will produce what? Decreasing the activity of the indirect pathway. So again, we have direct pathway for the initiation of voluntary movement that usually end by stimulation of the thalamus and stimulation of the cerebral cortex. We have indirect pathway to produce suppression or of the unwanted movement or termination of the voluntary movement, and this usually occur when the globus pallidus internal segment produce inhibition of the thalamus and the cerebral cortex. Dopamine produce activation of the direct pathway through the dopaminergic subtype 1. Dopamine through the D2 receptor, the dopaminergic subtype 2, produce decreasing the activity of the indirect pathway. And it's very important to understand the pathogenesis of Parkinsonism. Functions of the basal ganglia. The basal ganglia exert its function through the connection with the medial motor system and the lateral motor system. It has a connection with the corticospinal pathway and the retrospinal pathway, and these two pathways are concerned as the lateral motor system that control the activity of the distal muscles that produce the skilled movement, At the same time, it has a connection with the reticular formation that gives the reticulospinal tract, which is a part of the medial motor system controlling the activity of the axial muscles, that produce postural regulation or regulation of posture. And if we look to the function of the basal ganglia, include planning and programming of motor actions, executing pattern of learned movements, postural regulation, initiation of automatic movements, and regulation of muscle tone. These are the five main functions of the basal ganglia: planning and programming of motor actions, executing pattern of learned movement, postural regulation, initiation of automatic movement, and regulation of muscle tone. And the basal ganglia produce this action through its connection with the medial motor system and the lateral motor system. The lateral motor system that include the corticospinal and rubrospinal tract, and the medial motor system that include the reticulospinal tract. The first function of the basal ganglia is the planning and the programming of motor action. The basal ganglia produce something which is called the cognitive function, or the transformation of the thought into motor act, as a sequence of the connection of the basal ganglia with the association areas and the motor areas. So the basal ganglia receives input from the association areas that's informing the basal ganglia about the position of the body in relation to each other, and the position of the body in relation to the surrounding environment through the auditory association areas and the visual association area. And then the basal ganglia is going to detect the motor program by detecting when to move, where to move, and what muscles used or needed to produce this movement. So the cognitive function of the basal ganglia is not a function of the basal ganglia alone, but it's a function with the association areas, like the sensory association areas and the motor association areas. These provide information to the basal ganglia, and the basal ganglia put the motor program that will decide the transformation of the thought into act by detecting when to move, where to move, and what are the muscles used for the movement. And this is used by the connection with the association areas and the motor executive area. So again, the basal ganglia receives the information about the position of the body in relation to each other through the somatic sensory association area, and from the visual and auditory association area they are going to provide information about the position of the body in relation to the surrounding environment. Then the basal ganglia will put the motor program and send it through the thalamus to the motor areas that perform the action. So that is the production of the motor movement, or the planning and the programming function of the basal ganglia. The second function is the executing pattern of learned movement. Many patterns of learned or skilled movement, such as writing, typing, and many hand skills, in addition to certain aspects of speech, are considered as skilled movement and usually performed so rapidly that could not allow for sensory feedback control. It is produced without the need for the sensory feedback control, or before the sensory feedback control comes back to the cerebral cortex. It is believed that there is a specific pathways or specific circuits between the basal ganglia and the cerebral cortex have been established to perform these types of movement. The rapid skilled movement are established after several training period, and in this condition they have something which is established pathway between the basal ganglia and the motor cortex. Once you are going to perform this movement, the connection between the basal ganglia and cerebral cortex is starting, allowing you to perform this movement without the need of sensory feedback. Postural regulation. As we mentioned, the basal ganglia act as a bridge between the lateral motor system that produce the movement of the distal parts of the limbs and the medial motor system that produce control of the position of the trunk and the axial muscles. So usually the basal ganglia produce the appropriate or provide the appropriate positioning of the shoulder and elbow to be stable or to provide a postural background for the production of voluntary movement by the distal muscles of the hands. The basal ganglia provide an important bridge that links the activity of the lateral motor system that produce the skilled movement and the medial motor system that involved in the postural regulation. So the basal ganglia provide a postural background during the production of the voluntary movement. It has the connection with the lateral motor system that controls the activity of the distal muscles of the limbs and with the medial motor system that involved in the postural regulation. So usually the basal ganglia try to adjust the tone of the muscles of the trunk and the proximal muscles of the limb through its connection with the reticulospinal tract in order to provide postural support. In order to provide postural support. The next function of the basal ganglia is the initiation of automatic movements or subconscious movements like swinging of the arms during walking or the facial expression that appear during speaking. All these patterns of movement are thought to be produced by the basal ganglia, as we find in certain lesions like the Parkinson's or in Parkinsonism there is loss of the facial expression and the patient has something called mask face. The last function of the basal ganglia is the regulation of the muscle tone. As we mentioned previously, the output coming from the basal ganglia to the thalamus is inhibitory, and the thalamus is excitatory to the cerebral cortex. That means that the basal ganglia is inhibitory to the cerebral cortex, or the output coming from the basal ganglia is mainly inhibitory to the cerebral cortex, as it inhibits the thalamus, and the thalamus will consequently has less excitation to the cerebral cortex. At the same time, the basal ganglia produce inhibition of the reticular formation, so we can say that the basal ganglia inhibit the muscle tone. And if we look to the patient with Parkinsonism or lesion in the basal ganglia, they have something which is called rigidity or hypertonia or hyperreflexia. This means that the basal ganglia output mainly to the muscle tone is inhibitory. Disorders of the basal ganglia. The most important of these disorders is Parkinson's disease. Parkinson is the more common degenerative disease of the brain, and the primary lesion is usually a degeneration affecting the nigrostriatal fibers, leading to decrease in the dopamine secretion in the striatum. Most of the cases of the Parkinsonism are idiopathic, which is due to degeneration of the nigral dopaminergic neurons, while some causes may be related to the atherosclerosis or exposure to certain toxins and the prolonged intake of tranquilizers. So usually we have either idiopathic or non-idiopathic caused by repeated head trauma, the dopaminergic 2 antagonists, toxins and inflammatory, usually affecting the substantia nigra, leading to decrease in the dopamine secretion in the striatum. And usually it will be associated with the major disorders including static tremors, rigidity, and akinesia. Akinesia or bradykinesia, these are the three main manifestations of Parkinsonism: static tremors, rigidity, and akinesia. In akinesia, the voluntary movement is limited. Loss of the associated movement or subconscious movement, like the swinging of the arms during walking, is lost. And if we look to the speech, it is monotonous. Gait is shuffling gait. Usually, the disorders in the secretion of dopamine associated with cognitive disorders and mental apathy. That is the shape of the substantia nigra in normal persons, and that is the shape in Parkinsonism. The density of the neurons has been markedly decreased in the Parkinsonism. The direct pathway and indirect pathway in Parkinson's disease. In Parkinson's disease, with a decrease in the dopamine secreted from the nigrostriatal fibers to the caudate and putamen, decreasing the activity of D1 receptors and D2 receptors. This will produce a decrease in the stimulation of the striatum. When we decrease the stimulation of the striatum, less GABA will be secreted on the globus pallidus internal segment. So the globus pallidus become disinhibited or excited, leading to more release of GABA on the thalamus, more inhibition on the thalamus. This will lead to decrease in the excitation of the cerebral cortex, lead to decrease in the excitation of the cerebral cortex. With decreased excitation in the cerebral cortex, the patient will suffer from the akinesia, which is the most prominent manifestation in Parkinson's disease. Again, decreasing dopamine in the striatum, which is usually increase the activity of the striatum in the direct pathway. It will lead to decrease the release of the GABA on the globus pallidus internal segment, leading to the more secretion of GABA from the globus pallidus to the thalamus, more inhibition of the thalamus, less excitation of the cerebral cortex. So there is decrease in the excitation of the motor cortex, leading to the akinesia or the decrease in the initiation of the voluntary movement. And if the subject try to perform the voluntary movement, the movement will be exhausting, will be limited, and will be stiff. There is difficulty in the initiation of voluntary movement due to decrease in the excitation coming from the thalamus to the cerebral cortex. And if we look to the manifestation of Parkinsonism, there is rigidity with increase in the muscle tone in the flexors than the extensor. So usually the subject has the flexion attitude. There is akinesia or bradykinesia, inability to initiate the voluntary movement, with loss of automatic movements, shuffling gait, mask face, monotonous speech. The akinesia that occur in Parkinson's disease is often more distressing to the patient than are other symptoms. It is the most distressing symptom to the patient, more than the rigidity or the tremors, because to perform or the even simplest movement in severe Parkinsonism, the person must exert the highest degree of concentration. It needs a high degree of concentration to perform the simplest movements. The last is the resting tremors, which is static tremors, appears during rest and appear during voluntary movement. And these tremors usually exaggerated with emotional stress. Exaggerated with emotional stress. The second lesion is Huntington disease, or the chorea present in Huntington disease. In Huntington disease, there is damage in the caudate. There is damage in the caudate with decrease in the GABA released on the globus pallidus, decreasing the GABA secreted on the globus pallidus. Huntington disease is a hereditary disorder, usually begins at the age of 30 and 40. It is an autosomal dominant disease. It is characterized by the chorea movement, or the sudden movement that usually affect the one limb or a great part of the body. It usually appears as shaking of the hands or shaking of the shoulders. It is caused, as I mentioned, by the loss of the cell bodies of the GABAergic neurons in the caudate nucleus and putamen, and the acetylcholine secreting neurons in many parts of the brain. So usually it is associated with severe dementia. It is usually associated with severe dementia. So there is chorea present in the Huntington disease as well as severe dementia. And if we look to the direct pathway and indirect pathway in the Huntington disease, there is increase in the excitability of the motor cortex. There is increase in the excitability of the motor cortex, and this is usually comes due to decrease the inhibition coming from the globus pallidus on the thalamus. Decrease the inhibition coming from the globus pallidus on the thalamus. And this is usually caused by what? The decrease in the GABA released from the striatum to the globus pallidus. We have degeneration of the GABAergic neurons connecting the striatum with the globus pallidus. So the globus pallidus neurons become, especially the external segment, become disinhibited. And when these neurons become disinhibited, we can say that they are excited, leading to more release of GABA on the subthalamic nucleus, inhibiting the subthalamic nucleus, inhibiting the subthalamic nucleus. And this will decrease the excitation coming from the subthalamic nucleus to the globus pallidus internal segment. And when the globus pallidus become less stimulated, they cannot produce inhibition on the thalamus or decrease the inhibitory effect of the globus pallidus internal segment to the thalamus, decrease the inhibitory effect of the globus pallidus internal segment on the thalamus, so the thalamus can produce greater excitation of the cerebral cortex. Again, in Huntington disease, there is decreased GABA released from the striatum on the globus pallidus external segment. The globus pallidus become disinhibited, and when the globus pallidus external segment become disinhibited, it's going to produce inhibition of the subthalamic nucleus. Decreasing the activity of the subthalamic nucleus will lead to decrease its excitation effect on the globus pallidus internal segment, and when this occurs, we have decreased inhibitory effect coming from the globus pallidus internal segment on the thalamus, decreasing the inhibitory effect coming from the globus pallidus internal segment on the thalamus, so the thalamus is excited, leading to more excitation of the cerebral cortex. The next lesion is acidosis, which is usually caused due to extensive damage of the basal ganglia involving the striatum and the globus pallidus. It is characterized by a slow twisting movement affecting the hands and fingers. Slow twisting movements affecting the hands and fingers. The last lesion is the balismus, which is usually caused due to damage of the subthalamic nucleus. Damage in the subthalamic nucleus. It is characterized by spontaneous attacks of involuntary movement, spontaneous attacks of involuntary movements that's involving large muscles or large areas of the body, large muscles or large areas of the body, and this can occur and disturb the subject equilibrium. It usually appears as a sudden jerky flexion or extension of a limb, and usually can disturb the equilibrium, Non-motor functions of the basal ganglia. Non-motor function of the basal ganglia. We have another function to the basal ganglia non-related to the motor control or regulation of the voluntary movement, like the initiation and determination of cognitive processes such as planning, working memory, and attention. And if we look to the cortical input to the basal ganglia, it's coming from the dorsolateral prefrontal cortex, and then going to the anterior part of the caudate nucleus, to the globus pallidus internal segment and substantia nigra pars reticularis, to the mediodorsal, ventral, and anterior nuclei, mediodorsal and ventral anterior nuclei of the thalamus, and from the thalamus the fibers will go to the dorsolateral prefrontal cortex back, to the dorsolateral prefrontal cortex back, and this area are associated with the elaboration of thoughts and the formulation of ideas, as well as the consolidation of memory. And if we look to the second function, or the limbic loop, that started in the amygdala, hippocampus, orbitofrontal cortex, anterior cingulate gyrus, and temporal cortex, going to the ventral striatum, to the ventral pallidum, and then to the mediodorsal nucleus of the thalamus, back to the cortical targets, which include the anterior cingulate gyrus and the orbital frontal cortex. This loop play an important role in regulation of emotional behavior and motivation. So we find that the basal ganglia can produce non-motor function like cognitive function, as well as regulation of emotional behavior and motivation. The cognitive function, as well as memory and attention, will be produced by the prefrontal loop, and the emotional behavior regulation and motivation will be produced by the limbic loop. There are circuits present between the cerebral cortex and the basal ganglia, share in the process of planning and working memory, while we have another functions related to the regulation of emotional behavior and motivation produced by the limbic loop. These are considered the non-motor function of the basal ganglia. Non-motor functions of the basal ganglia. So at the end, the basal ganglia produce motor function as well as non-motor functions.